This is Chapter 6 on microbial growth. Microbes, just like all living things, have certain physical and chemical requirements for life. The physical requirements may be things in their environment, so things like temperature. So all organisms have an ideal temperature range at which they're able to live. Same thing with pH. There's an acceptable range of pH that certain organisms prefer. And osmotic pressure. So those hypertonic, isotonic, hypertonic solutions. So remember we said the hypertonic is a more concentrated solution. So it's going to draw the water out of the cells. Isotonic is the same concentration on both sides, so everything's stable and balanced. Hypotonic is less concentrated than in the cell. So inside the cell is more concentrated than the solution. So water will go into the cell to try to dilute that concentration, causing the cell to swell and potentially burst. Chemical requirements, so things like nutrients and chemicals that cells need for their metabolism. So things like carbon. Right? So all organisms are carbon-based. Some other nutrients like nitrogen, sulfur, hydrogen, oxygen, and some organic growth factors like proteins and amino acids. So when we talk about microbial growth, we're talking about an increase in the number of cells. So normally when you think about growth, you think about a child growing up or a puppy maturing into a dog. So the organism gets larger. Microbes, being single-celled organisms, obviously can't get larger, but they can increase in the numbers in their colonies. So colony is defined as a group of cells large enough to be seen without a microscope. And generally, they're derived from a single cell. So in lab, when we looked at our plates for microbes in the environment, and we looked at the different colonies that grew. So one important physical requirement for microbial growth is temperature. So all organisms have their own minimum, optimum, and maximum growth temperature. So kind of the lowest temperature that they're able to tolerate, an optimum temperature where they grow the fastest and are the healthiest, and a maximum growth temperature. So remember, temperature is important for metabolism, those biochemical reactions and those enzymes and their function. So we can classify some organisms based on their temperature preferences. So your psychrophiles are your cold-loving organisms. So they prefer things in the freezing range or even below freezing. So these are the organisms responsible primarily for food spoilage. So we can preserve the shelf life of our food a couple of days by refrigeration, but there are still some spoilage bacteria that can survive in the refrigerator temperatures. So your leftovers from dinner the other night are probably only good for a couple of days, and then you might want to throw them out. So these psychrophiles, I always think of them as the psychos that love the cold weather. So my minimum temperature would probably be around 50 degrees. Mesophiles, meso meaning kind of in the middle, so these are moderate temperature organisms. So around our body temperature range. So most of the pathogenic bacteria and bacteria that we deal with in our bodies and in our lab, and in this class would be the mesophiles. So they live at the same temperatures as we do, so we're going to interact with them a lot more. The thermophiles are the heat-loving. So these organisms prefer the higher temperatures. Psychotrophs, or the psychos, as I call them, grow between 0 degrees Celsius and 30 degrees Celsius. 0 degrees Celsius is 32 degrees Fahrenheit, so that's the freezing point. So again, these are the organisms responsible for your food spoilage. So some bacteria can still survive and may grow in these refrigerator temperatures. So this chart is showing the surface area or the amount of food in a container and its effect on cooling and chance of spoilage. This larger container takes much longer to reach the refrigerator's ambient temperature than the smaller, more shallow container. So this darker band shows kind of the danger zone range. So the longer our food sits in this temperature range, the more the bacteria have a chance to grow and spoil our food. So the smaller container only spends less than an hour, maybe half an hour, before it reaches that cooler temperature. Whereas our larger container, right, is in this danger zone for several hours. Thermophiles' optimum growth temperature is between 50 to 60 degrees Celsius. So 60 degrees Celsius is about 140 degrees Fahrenheit. So the human body temperature is 37 degrees Celsius, so 98.6 degrees Fahrenheit. So these thermophiles in the 140 degree Fahrenheit range are found in places like hot springs and organic compost. The hyperthermophiles found in even more extreme temperature ranges nearing 200 degrees or more. pH is another important physical requirement for microbial growth. Most of our bacteria prefer to grow between pH of 6.5 and 7.5. Molds and yeasts prefer a more slightly acidic environment, pH of 5 and 6. The acidophiles, or acid-loving organisms, grow in the very acidic environments. Osmotic pressure is another physical requirement or environmental factor for microbial growth. So in hypertonic environments where we have a higher solute concentration outside the cell than inside the cell, will cause plasmolysis, or the cell to essentially shrivel up and die. So the water will be drawn out of the cell toward this more concentrated solution due to that high osmotic pressure. However, some organisms prefer these super concentrated or hypertonic environments. So extreme or obligate halophiles require that high osmotic pressure or high salt environment. Facultative halophiles can tolerate high osmotic pressures if necessary. So sometimes when we talk about obligate versus facultative, obligate means that they are obligated to be in those high salt environments. Right? So if you're obligated, right, there's no way out of it, you have to do it. So I always use the example, I'm obligated to spend Thanksgiving with my in-laws every year. It doesn't matter if I want to or not, I have to go. Facultative means that it's flexible. So if the situation arises, right, where we encounter a high osmotic environment, right, the cell can tolerate it and be okay. One of the most important chemical requirements for all life and microbial growth is carbon. So all living things are based on carbon chemistry. So if you ever take an organic chemistry class, it's nothing but carbon-based chemistry. So carbon forms the structural backbone of all the organic biological molecules. So proteins, sugars, fats, everything is based on carbon. Some organisms, like chemoheterotrophs, use organic molecules as energy. So we acquire this carbon from a food source and reconfigure the carbon into other biological molecules. Autotrophs use carbon in the form of CO2 for photosynthesis. So here we have glucose, a sugar. So it's a 6-carbon sugar. Amino acids that form proteins are carbon-based. Fatty acids are just chains of hydrocarbons. ATP, adenosine triphosphate, the primary energy molecule. So carbon's important for organic chemistry because it's able to form large, complex, and diverse molecules. So when we talked about chemical bonds in anatomy and physiology, remember that octet rule, or the magic number that all atoms want to have, is 8 valence electrons. So they interact with other molecules or atoms to reach that goal of 8. So carbon is able to form 4 separate bonds and share up to 4 electrons. So it could form 4 single bonds. Right? So this carbon here has a bond to another carbon, and to 2 hydrogens. It could form 2 single bonds and a double bond. So now we have kind of this kink in the chain. Not shown here, it can also form a triple bond. So it would be triple bonded to 1 atom, and single bonded to another. So we have all these various shapes and functions of biological molecules. Nitrogen is an important chemical requirement for proteins, DNA, and ATP. Some bacteria use nitrogen for nitrogen fixation in their metabolism. So all of the bases, the letters in DNA, the A, T, C, G, are sometimes referred to as the nitrogenous bases because they contain nitrogen. So nitrogen is a vital component in DNA. Nitrogen is also important for the production of ATP, that all-important energy molecule. Sulfur is used in some amino acids like methionine, cysteine, and some vitamins like thiamine and biotin. Some bacteria, like your purple sulfur bacteria, are able to decompose protein for the sulfur from these amino acids. Phosphorus is a very important chemical requirement. It's used in DNA, RNA, and ATP, as well as form the basis of the cell membrane. So remember, the cell membrane is a phospholipid bilayer. Phospholipid has a phosphate, right, or phosphorus attached to that fatty acid chain. Right? It's also going to form the P in ATP. So adenosine triphosphate has those 3 phosphate groups. Trace elements are things like vitamins and minerals, or inorganic elements that are only required in small amounts. So most of these are going to function as your enzyme cofactors, so things like iron and zinc. So remember, the cofactors or coenzymes, we said, allow an enzyme to function. So without the coenzyme, our substrate won't fit on our active site. So we need the coenzyme to allow that enzyme to function. Well, oxygen is an important chemical requirement for us. Obviously, we cannot live without oxygen, but different microbes have different tolerances to oxygen. So obligate aerobes, so again, obligate meaning they have to, so these can only grow in the presence of oxygen. So oxygen is required. So we humans as well are obligate aerobes. We have to breathe oxygen or we will die. So these are noted in tube growth, so they'll grow near the top or the surface. So this is where the oxygen is in the test tube. Facultative anaerobes, remember facultative meaning they can adapt, they can tolerate different scenarios. So these microbes can tolerate both aerobic and anaerobic, but they will grow greater in the presence of oxygen. So when we look at these in the test tube, you'll see most aggregated near the top, near the oxygen, but there are some down below in the aerobic environment that are still able to grow a little bit. Obligate anaerobes can only grow without oxygen. So oxygen is actually kind of a toxic gas to these obligate anaerobes. So you'll see these grow at the very bottom of the tube, farthest away from that oxygen layer. Aerotolerant anaerobes are similar to facultative, but they don't really have a preference. The facultative prefer to have oxygen, but they can deal without it. Aerotolerant can only have anaerobic growth, but they can tolerate oxygen, so they'll still grow in the presence of oxygen. And microaerophiles have only aerobic growth, but they require lower concentrations of oxygen. So they're not completely anaerobic at the very bottom of the tube, but they're not completely aerobic where they need the full force of the oxygen in the air. So they grow about halfway down where the oxygen just starts to dissipate a bit. So here's just a fun fact and maybe a potential bonus question on your next exam. So there was no free oxygen in the early Earth atmosphere. So when life first evolved, the first cells evolved and their metabolism evolved, there was no oxygen in the environment. So there was no oxygen-based metabolism. So this is why we still have some anaerobic organisms today. Why wasn't there any oxygen? We have lots of oxygen now, so where did all of this oxygen come from? The answer is photosynthesis. So free oxygen in the early Earth atmosphere did not accumulate until the evolution of photosynthesis. So once those photosynthetic bacteria started to photosynthesize. Remember, oxygen is a waste product of photosynthesis. All of that oxygen started to accumulate in the atmosphere. And originally it was considered kind of a toxic, poisonous gas to these cells. They had no way to deal with it. But through evolution, the cells adapted and figured out a way to use the oxygen to their advantage. Organic growth factors are organic compounds obtained from the environment or other organisms. So basically the building blocks for the biological molecules, amino acids and vitamins, nucleotides. So amino acids are the building blocks for all proteins. So we can make every protein imaginable by arranging these 21 different amino acids in different configurations. So a lot of these building blocks are recycled. So when you eat a steak for dinner, right, you're essentially eating muscle tissue from a cow, which is protein. Right? So your digestive system will break down that protein in the steak to the basic amino acids. And then your body will use those amino acids to make your own muscle tissue protein, whatever other protein is needed by the cell. Biofilms are microbial communities. So we said before, microbes don't generally live in pure isolated colonies. They live in these communities with multiple microbes all kind of living, sharing the same space. So these biofilm communities form slime or hydrogels that help them adhere or stick to hard surfaces. Once the cells reach high enough density, so there's enough cells in the population, they can start to communicate via quorum sensing. So in quorum sensing, essentially the bacteria can talk to each other. So if you only have a few cells, right, there's not as many recipients for your signals or your messages to go to. Right? We have lots of cells in the same space, right? It's much more likely that your signal will get spread to more cells. So these bacteria will secrete an inducer or a signaling chemical to help attract other bacteria cells to the community. They basically put up a houses for sale sign. So these communities kind of work together and they can share nutrients and they help to shelter other bacteria from harmful environmental factors. So overall, these biofilms can be more resistant to treatments like antibiotics. So the cells deeper in the biofilm in the lower layers would be more protected against any antimicrobial agent we try to apply to it. This is actually a figure showing the different types of bacteria on a normal tooth surface. So the reason we have to brush our teeth every day is because these biofilms can start to form. So when your teeth get that kind of furry, fuzzy feelings, you need to brush your teeth, that's that biofilm forming. So we have some early colonizers that are kind of the first on the scene to kind of set the stage for the new community. And then we have some later colonizers. So if we don't brush our teeth quick enough, we have enough time for new bacteria to move into the neighborhood. So when biofilms get large enough, they can form kind of a primitive circulatory system. So they'll form these channels in different clumps of the biofilm. So as fluid or the water current flows through, it can deliver nutrients and remove the bacterial waste products. So again, essentially kind of like a very early primitive circulatory system. They can also send off new migratory clumps to start new biofilms elsewhere. Culture media is what we use to grow bacteria on in the lab. So these are specific nutrients prepared for microbial growth. Culture media should always be sterile, meaning it should not have any living microbes on it when we first use it. When we inoculate a sterile culture media, right, we're introducing microbes into that medium. So a culture would be the microbes that grow in or on our culture media. So when we do lab and you do your inoculations, you had a culture, so a broth culture or cultures on Petri plates that you took a piece of or a sample from and then inoculated or introduced to a new media. Solid culture media uses agar, which is a complex polysaccharide harvested from red algae. It's used as the solidifying or the gelling agent for our petri plates, our slant tubes, and our deep tubes. It's good to use the agar from the red algae because it's generally not able to be metabolized or broken down by any microbes that we deal with. Red algae agar is also used for some Asian desserts and a thickener in soups and ice cream. A chemically defined media is one where we know the exact chemical composition in that media. So in this chemically defined media example, right, so we know exactly how much of each nutrient or component is in defined medium. Complex media includes extracts or digest down products of things like yeast, meat, or plants. So depending on the type of media, the type of culture we're trying to grow, chemical composition can vary from batch to batch. But there's two basic forms for complex media. So we can have a nutrient broth, which is just the liquid portion without the solidifying agar, or a nutrient agar. So we do have that red algae agar added, right, to solidify it, kind of like Jello, right? So we can pour it on a plate and have a large surface area for growth. We could have a solid or a deep tube, right, the one that we stab down into to check for motility, or the agar slant, right, just kind of a smaller version of the plate just to see if it grows on a solid surface. We're also able to culture some anaerobic bacteria with special types of growth media and methods. So anaerobic growth requires a reducing media. So it contains special chemicals like sodium thioglycolate that will combine with oxygen to deplete it. So this is called a thiotube. So it has this indicator here to show that it has not been exposed to oxygen. This is an anaerobic chamber, sometimes called a brewer's jar. So we would put our plates in this jar, this chamber, and have special CO2 packets that will cause this to be a anaerobic chamber. And if you want to get really fancy, we have airlock containers with the special arm ports for these anaerobic environments. So depending on the biosafety level of the type of organism you're working with, we can use different special culture techniques. So BSL-1 is kind of the most basic, the safest level. So no special precautions, just basic teaching labs. BSL-2, you need lab coat, gloves, and eye protection. So this would be our level in our lab. Everything higher than that, you need more specialized equipment, biosafety cabinets to prevent airborne transmission, or special HEPA filters to filter the exhaust air for certain airborne diseases. We can use different types of culture media to select for the growth of certain bacteria or to help differentiate between different bacteria. Selective media is, think of in terms of natural selection. So the natural selection of this media is going to suppress or select against the microbes that we don't want, and it's going to encourage the microbes that we do want. So sometimes these will contain certain chemicals that inhibit or suppress growth of every other type of organism. So one type of selective media that we'll see in lab is EMB, stands for eosin methylene blue. So this is selective for gram-negative bacteria. Methylene blue dye, the same one that we used in our simple stain, in this media is going to prevent the growth of gram-positive bacteria. So EMB is selective for gram-negative. Right? So this is E. coli on an EMB plate. So these are really cool. They grow this really pretty metallic green on EMB. Differential media allows us to differentiate or distinguish between colonies of different microbes on the same plate. Some media can have both selective and differential characteristics. So this is an example of a blood agar plate. So it's enriched differential medium. So it allows us to distinguish between organisms based on how they undergo hemolysis. So how they react to the blood cells on these plates. So Streptococcus pyogenes can be identified by a beta hemolysis, so just a complete clearing of the blood cells. So it's rupturing the blood cells right? So that's why we have this clear zone. Streptococcus pneumoniae is characterized with alpha hemolysis. So we have this kind of greenish color to the agar. Enterococcus faecalis has no hemolysis, so it has a gamma classification. Right? So it doesn't break down the blood cells. It's able to grow on them, but it doesn't break them down. Another selective and differential media is MSA, or mannitol salt agar. So it's selective because of the salt, right? So only Staphylococcus species can grow in this high salt media environment. It's differential because Staphylococcus aureus will ferment the mannitol sugar in the media and turn it yellow. Staph epi does not ferment mannitol, so the agar will stay pink. And then this is a non-Staphylococcus species, so it's selected against, and it cannot grow on this plate. So remember we said sometimes the scientific name of an organism can be kind of descriptive or some of its characteristics. Right? So Staphylococcus are those spherical shaped cells in clusters, and aureus means gold. So Staphylococcus aureus turns our MSA plates gold. Enrichment cultures just help to encourage the growth of a desired microbe. So we kind of pinpoint what's its favorite nutrient, right? And we increase those particular components to kind of tailor it to the taste of our microbe of interest. So we want to grow those microbes up to a detectable level. When we talk about pure cultures, we're talking about only one species or strain. So a colony is a population of cells that arise from a single cell, typically on our Petri plate. Sometimes you'll see the colonies referred to as CFUs, or colony-forming units, because technically they are millions of cells, right? But they were all derived from a single unit cell. So we use the streak plate method in lab to help isolate these pure cultures. So if you have a mixed sample, right, so we have two bacteria mixed together. So when we do the streak plate, you streak your first streak. Then you sterilize your loop, or you get a new loop, and you streak again. So we're taking some of the cells, right, from these streaks here, and we're just kind of pushing them over to this side of the plate now. Okay, so then we would sterilize our loop, get another loop, and then we take some of these cells, a couple cells from here, and push them over there and spread them out. So ultimately, we'll see a progressive thinning, right, of our bacterial growth until we have these single isolated cells. So now we're able to distinguish between the two different species, the red ones and the yellow ones. Where up here, where we have a lot of cells, a lot of growth, it's hard to kind of separate and see what's what. Right? So we spread it out thinner and thinner and just streak it out, right, to get those isolated colonies. Bacteria divide by binary fission, meaning they essentially split in half and form copies or clones of themselves. So again, remember, microbial growth, we're talking about an increase in the number of cells, not the size of the cells. So the cells will stay pretty much the same size, they'll just increase in their numbers. So the process of binary fission begins with replicating the DNA in the nucleoid. Right? So we have to make a copy of the DNA to give to our copied new cell. Then the plasma membrane and the cell wall will start to kind of pinch off in the middle here, start to form a new cell wall. Right? So eventually the new cell wall is completed. So we've separated these two cells, and now they separate. So because bacteria undergo binary fission, they can increase their numbers very rapidly and exponentially. The generation time is the time it takes for a cell to divide. So this generation time can range from 20 minutes to 24 hours. And again, the binary fission is going to double the number of cells every generation. So because after just a few generations, we start to deal with exceedingly large numbers, growth curves are usually represented logarithmically. So in this example, we have one bacteria cell, right, that starts our colony, so our founder. right? So it multiplies into 2 cells. Right? 2 cells multiply into 4. 4 becomes 8. 8 becomes 16, and so on and so forth. Right? So after only 20 generations, right, we're up to 1,000,000 cells. So if a bacteria's generation time is, say, 1 hour, so in less than a day, less than 24 hours, we already have over 1,000,000 cells. So if we try to graph this on a standard graph, we have this really sharp uptick in this exponential growth. So it would just get really hard to keep graphing it this way. So that's why we use the log scale. So a true exponential curve on a logarithmic scale would be a nice linear slope. So another potential bonus question on your next exam, a little riddle about exponential growth. So a lily pond starts with a single leaf. Every day the number of leaves will double. So 2 leaves on the second day, 4 leaves on the third day, 8 leaves on the fourth day, so on and so forth. On day 30, the pond is full. There's no more room for any more lily pads. So on which day was the pond only halfway full? On the 29th day. Right, so remember, we're doubling every day. So the first, right, almost 3 weeks or more, right, we're just dealing with small numbers. So 2 double is 4. So both are small numbers, but you get increasingly larger numbers. And once you start to double a large number, it becomes a larger number, right? And then we have kind of this exponential growth, this kind of shooting up. So it's not until the 29th day that the pond is only half full. Most bacterial populations follow a standard kind of pattern or phase of growth. So the first phase is the lag phase. So during this phase, so looking at just the sheer number, the log number of bacteria in our population, right, it's pretty low, right? There's no real increase in population. The new settlers have arrived. They're kind of getting their bearings. They're preparing for their population growth, right? Getting all their nutrients and enzymes in order. So the log phase, or the logarithmic or exponential phase, is where we have this kind of taking off in the population numbers. So they shoot up very rapidly, right, exponentially. The stationary phase is the period of equilibrium, right, where the line is kind of stationary, meaning it doesn't go up or down. So during this stationary phase, the number of microbial deaths is balanced by the production of new cells. So cells are dying at the same rate that they are being reproduced. So we're just replacing the ones that die, right? But we're not increasing our overall numbers. And finally, the death phase. So now the population will start to decrease. So now cell death is outpacing cell reproduction. And it will also decrease at a logarithmic rate, so at that same kind of exponential rate, just in the other direction.